Okay, so we're going to go ahead and try this lecture, starting where we stopped the other day. We're talking about synapses in class. Synapses convert electrical signals to chemical signals. To be honest, electrical signals are really just chemical signals anyway, as we'll see in a little while. So as the action potential comes down the axon, it gets to the synaptic end bulb, or some sort of synaptic ending. It doesn't have to be an end bulb. As an example, autonomic nervous system endings like the sympathetic system have what they call varicosities, which are like little beads on a string that all individually release neurotransmitters. So it has multiple endings and a much more diffuse effect then because of that. So those endings contain neurotransmitter. That neurotransmitter could be a number of different types of chemicals. The neurotransmitter gets released once the action potential comes down the axon and causes calcium. That's the number 2 there, the Ca+2. See the number 2 calcium channels to open, because calcium is higher concentration on the outside of the cell than inside of the cell. Calcium diffuses into the cell. This provides the energy to cause the synaptic and end bulb to release neurotransmitter into the synaptic cleft. The neurotransmitter travels across the synaptic cleft to the postsynaptic membrane, where it typically binds to some sort of ligand-gating ion channel, and that causes an action on the other side. So that's a fairly general view of what a synapse looks like or does. Synapses literally mean to connect. It's the point where an axon meets some sort of target. That target could vary. It depends on the synapse itself. It could go to another neuron, the dendrite, axon, or cell body. It could go to a non-neuron, such as a gland or muscle. So there's a lot of different places we could have. As mentioned previously, a presynaptic cell releases the signal, and the postsynaptic cell receives the signal. That generally has a receptor, which then carries out the effect. There are 2 types of synapses that we see in the human body. One type is much, much more common. The electrical synapse is very rapid, but has very little control, while the chemical synapse, which is by far the most numerous one, is slower, but has much more control as well. The electrical synapses have the direct transmission of electrical signals that go from one cell to the other. Those occur via gap junctions. It's a very, very fast connection. It helps to enable certain synchronized activities. We see this in the brain occasionally, but the problem is a couple. One, most of the electrical synapses are bidirectional, which means the signal can travel in the wrong direction, and we have very little control over an electrical synapse, and oftentimes at the expense of certain things. In the case of electrical versus chemical signals, speed, we often favor a more controlled output with something like a chemical signal. So the chemical signals can work slower. The synaptic delay of a chemical signal is about half a millisecond, so 1/2000th of a second. So that's the extra time it requires to have a chemical signal. So it is slower, but we can regulate the signal transmission through a number of different ways. One, we can assure unidirectional flow. That means that we're going from the synaptic ending to the postsynaptic cell, and receptors do that because the only place that we will have an effect is where we have a receptor. So that helps. Of course, that slows it down a little bit as the neurotransmitter has to be released from the synapse move across by diffusion and then bind the receptor and then carry out the response. So it does slow things down a little bit. The other thing we can do also is we can help modulate that signal by changing the number of receptors. In the homeostasis section, we talked about upregulation or downregulation, where we can change the sensitivity or number of receptors and modulate the response somehow, increase it or decrease it depending on the conditions. If we have that unidirectional flow, then that helps to ensure what we call orthodromic conduction. So orthodromic conduction is when we go from the cell body, so here's orthodromic down here, from the cell body to the axon, that's the normal direction, and it prevents what they call antidromic conduction. Antidromic is when we go in the reverse direction, axon to cell body. That generally wouldn't be good, and it would disrupt any signals coming from the other end. So signals need to go down the axon in a single line. A couple other things that we can look at, but the two big things are the speed and control. Even though chemical synapses are a little slower than electrical synapses, they're still very fast, and the control and specificity allows us to favor chemical synapses much more than electrical synapses. We see very few electrical synapses in the human body. As we discussed again in the homeostasis section, in order to shut off a signal, one of the things we have to do is we have to remove the neurotransmitter. The neurotransmitter can be removed in a number of different ways. One is active removal through a pump, some sort of pump. So that neurotransmitter we see here in number 1 would be pumped either into a glial cell or pumped back into the membrane where it's stored. We can have active degradation, that's number 2. That's an enzyme that breaks the neurotransmitter down so it's no longer active because it's no longer specific for the receptor. As an example, we see that in different types of synapses that use acetylcholine, and they break down by acetylcholine esterase. It's an enzyme that sits in the synaptic cleft and inactivates acetylcholine. The third way to do it, right up here, is you can just diffuse away. Here it's showing diffusion in the interstitial fluid, or just diffusion in the bloodstream and potentially acting as a hormone now because it's a chemical that's in the blood that would potentially have target cell receptors that it can bind to. The fourth way, which isn't really shown on this figure too much, is we can change the target receptors themselves in terms of changing their number, sensitivity, in downregulation, or just completely taking the receptor and the neurotransmitter complex and moving it into the postsynaptic cell. So we could do basically receptor-mediated endocytosis would also help to shut off the signal. But the three that are mentioned are probably the most common ways that we see that. The other thing to remember is that neurotransmitters can often be recycled. One of the most common neurotransmitters in the human body, in fact it is the most common, is acetylcholine. Acetylcholine is made fairly easily within the cell. If you take acetyl coenzyme A, which we saw in metabolism, we can make it various number of ways. We can make it from glucose, we can make it from lipids, we can make it from proteins by converting them into acetyl coenzyme A through ketogenic amino acids, or we could make them into glucose, which then can turn into acetyl-CoA by glucogenic amino acids. So, acetylcholine is pretty common in the cell. Choline is a fatty acid, so all we need to do is take that acetyl coenzyme A, hook it up with a choline molecule, and make acetylcholine, and then we can release it into the synaptic cleft as a neurotransmitter. That enzyme I mentioned before, acetylcholine esterase down here, can take the acetylcholine and inactivate it by basically chop it in half, and now we're left with an acetate molecule and a choline molecule. Since the acetate molecule, if you just put a coenzyme A on it, becomes acetylcoenzyme A, that's pretty common in the cell. We don't really need to worry about recycling it or taking it back in. But the choline is much less common, and so what we want to do with the choline right here is we'll actively reuptake the choline, transport it back to where we can make it, and then use an enzyme to make acetylcholine again from the acetylcoenzyme A's we can get from mitochondrial activity, as we saw in metabolism. So that's one way to do it. Norepinephrine is also recycled this way, and there are various enzymes within the synapse that will help break norepinephrine down and inactivate it as well. There are a number of different neurotransmitters and a wide variety of chemicals that they would respond to. Here's just some examples, kind of combined stuff. So nicotine is what we call an acetylcholine agonist. So it excites. Remember, agonists are chemicals that excite receptors. So an agonist for acetylcholine would excite acetylcholine receptors. Nicotine does that. We have the endocannabinoid receptors that we can see in the body. That's what cannabis, basically like marijuana, right? THC would bind to and have an effect. There are serotonin antagonists, so those are chemicals that would block serotonin, and as an example of that would be LSD. We have histamine antagonists like Zantac and Pepsid AC, which is a slightly different chemical, but still it blocks histamine receptors, and that's like ranitidine. And even alcohol is what we call a GABA. GABA is an inhibitory chemical we see in the brain, and basically it was called a GABA potentiator. So you have all these different chemicals. We're not going to look at those at all in terms of class for the most part. We're going to focus primarily on two, especially when we get to the autonomic nervous system. Those would be acetylcholine and norepinephrine, which are the two most common ones we see in the peripheral nervous system. One of the other factors that kind of doesn't show very well in some of these diagrams we have is that all protein synthesis for neurons occurs in the neuronal cell bodies. In terms of the axons, they lack the protein machinery. They don't basically have any rough endoplasmic reticulum to be able to make the proteins that are released as neurotransmitters. So those have to be made up in the cell body. As a reminder from anatomy, the proteins are made in the rough ER or the RER, which are called Nissl bodies in neurons, or sometimes known as the chromatophilic substances. But basically those are in the cell body. And in order to get to the axon to be released, to the end of the axon, they have to be transported from one place to another. And sometimes that's a fairly long trip. As an example, if you look at the neurotransmitter that would be made for a somatic motor neuron that went to your big toe, the cell body for that would be up in the spinal cord where the proteins are made, but the synaptic end bulb would be in your big toe, essentially, which in some people, depending on height, could be 3 feet long. And so how do we get the neurotransmitter that's made in the cell body all the way down to the synaptic end bulb? And we'll talk about how that transport works. So axonal transport is designed to move materials from the cell body to the synapse, and in some cases from the synapse back to the cell body. If it moves from the cell body to the synapse, it's called anterograde. So anterograde means that from the cell body to the synapse. If it's going the opposite direction from the synapse back to the cell body, it's called retrograde. And retro almost refers to backwards or back in time. So that kind of gives you the idea there for that. There are two ways to move materials. One is called fast axonal transport. Fast is a relative term. In fast, the movement is about 400 millimeters a day, which, if you do the math, is about 16 inches a day. So it's not really very fast, but compared to the slow, it's significantly faster. This uses a overall branch of molecular molecules called kinesins. Actually, technically in one direction, in the anterograde direction, kinesins move substances from one to the other. Moving back, they're often called dynins, but we're just going to call them all kinesin. Kinesin, remember, kinese means movement. The N refers to a protein. So kinesins are just basically movement proteins. And kind of think of them as little railroad tracks within the neuron itself. So down here, well, I guess it's up here at the cell body, where we have the Nissl bodies, and you can see the representation of the rough ER and the Golgi. So we make the neurotransmitter in the rough ER. That's transferred to the Golgi, and then the Golgi spits out the little membrane packet with the neurotransmitter in it. It moves down the axon in the anterograde direction from the cell body to the synaptic end bulb via these kinesin molecules. They're active transport mechanisms that require energy. It unloads some of its substances at the end and then picks it back up if they're going to move anything back, and goes back and delivers substances potentially back to the rough ER or the Nissl body, or whatever terminology you want to use. So, as an example, if we looked at acetylcholine would move in the anterograde direction, while the choline, after the acetylcholine is broken up in the synaptic ending, then will reuptake the choline and send it back through fast axonal transport to be reassembled back into acetylcholine. So this type of movement, the fast, is bidirectional, and primarily the cargo tends to be neurotransmitter material, whether it's a neurotransmitter or the portions of the neurotransmitter to bring back and reassemble. It doesn't matter, but those are what typically happens there. The other way to have axonal transport, that is transport on the axon, is what we call slow axonal transport. It's only about 2.5 millimeters per day, which is about 1/10 of an inch. Just to kind of look at the relative changes, if you took that person that had a 3-foot axon from the back on the spinal cord to the big toe, and you use fast axonal transport, it would take a little over 2 days to move material from one end to the other. For slow axonal transport, it would take almost an entire year. So there is a very big difference between that. For the difference, we don't use protein. It's a passive transport mechanism for slow. We use what's called the axoplasmic flow. That is a gradient set up by the pressure difference between the cell body and the synaptic ending. And the cell body generally has a higher pressure, so movement of substances go from the cell body in the anterograde direction to the synaptic ending. So that is unidirectional. And the cargo tends to be enzymes and axoplasm, which is just the cytoplasm within the axon, and other materials that wouldn't be needed in an immediate fashion. So that's the difference between fast and slow axonal transport. So we're going to stop this section here and start with a whole new video series looking at the actual sodium-potassium pump, and review that a little bit, and look at some of the ion and ion concentrations, which really are what the crux of a large portion of the physiology within the nervous system really does.